Bismillahirrahmanirrahim. How are you all, dear students? Assalamu alaikum wa rahmatullahi wa barakatuhu. I welcome you in Pakistan International School Types virtual learning classes, and this is class for subject biology for second year students. Dear students, we have started chapter number 23 in which we are discussing chromosome and DNA, which is a very important part of our life or of our cell, of our nucleus, because it is carrying the hereditary material. In the previous lecture, we have talked about that chromosome or DNA, because DNA makes the chromosome, and chromosome are hereditary material, and they transmit the characters of one cell to the other, as well as the characters of one offspring to the other offspring. In that, we have just talked about uh, some experiments which have proved that DNA as the genetic material, because in the start, the scientists didn't know about that DNA, whether the protein which are present in the chromosome, they are the hereditary material, or DNA itself is the hereditary material. So it was proved by certain experiments which we have seen. Very important was Griffith's experiment, then Avery, McLeod, and McCarthy experiments. We have studied about them, and then we have just talked about the experiment and the result of Hershey and Chase. In the final result was that DNA is the hereditary material and not the proteins which are present over there. In today's topic, we will talk about the next step of DNA, that how that inheritance is shifted from one cell to another, or one offspring to one generation to another generation. That is through DNA replication. What is DNA replication, why it is important, and what is its mechanism? We will talk about this topic in detail. So let's get started. DNA replication, actually, that is the basis for inheritance. That is the basis for inheritance. And as we know, that it is the fundamental process occurring in all cells for copying DNA to transfer the genetic information to the daughter cells. Like it has to be copied to another copy and to transfer to another cells. So DNA first, for transferring its genetic material, it has to be duplicated, it has to be copied. That process is called DNA replication. So each cell must replicate its DNA before division. We know in mitosis and meiosis, we have seen in detail about this process, but right now we will take the different steps of DNA replication. Before that, we will see that DNA replication, how the scientists know that the DNA replicates and how does it replicate. That is very important. So as we know that the authentic and valid structure of DNA was given by Watson and Crick, that is the DNA is a double helical structure. And this double helical structure how it is replicated, because it is having two strands, double-stranded structure. So there are three hypotheses which are given by the studies of different scientists. So that is conservative replication, dispersive replication, or semi-conservative replication. They had to be hypotheses, they said to be models. So you can see here conservative replication in this step, then dispersive replication and semi-conservative replication. What are the differences between them? Let's talk about them in detail. That conservative replication. So what happened in that? Replicated DNA results in one old double-stranded DNA and one new double-stranded DNA. It means that DNA is double helical. We know that. But this whole strands make its complete replica. This method is explains that, for example, this one is the old one, and it just when it replicates, it founds complete both of the strands, the new one. It is called conservative, or the old one. It means the whole of the DNA is old and it makes its whole new copy with double stranded. So it is, we can say, the least acceptable model of DNA replication. Then we see the next one, dispersive replication. It means that replicated DNA results in each double strands with segments of old and new strands of DNA. For example, this is the old strand. So when we just make the new strands DNA mix, so what areas it's having, both of the strands are having some of the strands which are the new one, and some are, you can see here, some are like, we can just make it like this, they are the new one and some are the old one. That is also like the DNA is dispersed here in different segments. So that is also not acceptable by the scientists. But when the experiments were done about that, so the most acceptable hypothesis or model is semi-conservative replication. In that each double strand has one old strand and one new strand. For example, this one, what is it? It is the old one. So when it will make, or it will replicate, it will unwind, and then this is the old one and the new one will formed with this one. And here also the old one and one new copy of this will be formed. We will see this in the next diagram. So you can see this semi-conservative, conservative, and dispersive models in this diagram, and you can see it, understand it properly. This is the parent one. When it replicates, both of the strands get separated and or unwind, and they will form this blue one is the replica of this one, like the new copy of the old strand. So one is the template and the other is the new strand in first replication. In second replication, this one is the old one and this one is the new one. So this one is the old one and this one is the new one. So it means one of the strand is the old and one is the new one. That is the most acceptable model, and that's called hybrid molecules are formed in this situation. So if conservative model, we take that. So we'll see that parent, when it replicated, this is the old one and exact copy of both of the two strands will be formed. That is the new one. So by this one is the old one and this one is the new one. So it means from here, completely other than new one is formed, and like this it is happened here. In dispersive, you can see some of, like the pink is the old one and the blue is the new one. Oh, pink is the old one and blue is the new one. It means some fragments are the new and some fragments are of old DNA molecule. So this is the least acceptable model. To um, like to prove the DNA replication is semi-conservative, or actually the how the replication of DNA replication is done, uh, while in which type of method, so or in which type of model. So Michelson and Stahl experiment was done. Michelson and Stahl, what they did, they in 1958 in the California Institute of Technology, they done this experiment and they got a Nobel Prize after this discovery, that they evaluated that which type of model is the DNA replication model. So Matthew Michelson and Franklin Stahl, they have performed a very, very important and very interesting and good experiment. So that is the authentic experiment in recognition of the DNA replication model. So they designed the most beautiful experiment in biology, and they test the DNA replication models, and they used bacteria to grow in the heavy isotope of nitrogen. So what is their experiment about? That what they did, first, they grew bacteria in a medium containing N15. You know that N is having 14, its atomic weight. So they used N15 that is heavier in their experiments, and the bacteria are grown in the lab, and for many generations they provided their nutritional medium N15. And we know that N, nitrogen, is the major component of DNA, like deoxyribonucleic acids. So nitrogen is the major component of DNA. So that's why they just um, give in their medium N15, that is heavier. So what happened? All bacteria, they got N15 in their DNA, because in the many generations DNA repeatedly replicated their DNA. So they got in their DNA N15. So then in number second step, they transferred some of N15 bacteria to grow in N14 bacteria. Now they just shifted these bacteria into another medium in which they gave N14 media, N14, and they allowed them to grow for several generations. So here the DNA of newly divided bacteria will have N14 instead of N15, because they're taking from the medium. Okay, so what he did? He did when 3 DNA samples were taken from bacteria shifted from N15 to N14 medium. So he took some samples, how many? 3 samples of growing bacteria in N14 here, N14, because he shifted from N15 to N14, and then there's bacteria, sorry, N14. He just took the samples after different times, at different times, and studied the density of DNA of the cells via centrifugation. Okay, first he took first sample when he just put their sample in N14, and soon after that, yes, he took that some of the bacteria from them. He did not give any time, they did not give any time to the bacteria to replicate. So what happened? That is called, we say them at 0 minute, and that was the first sample. Okay, just he took 3 samples from that. One bacteria growing in N15, all DNA. Okay, so that was the result actually. So first at 0 minute, and then he took the other sample of some bacteria after 20 minutes, because after 20 minutes some bacteria have replicated. So when bacteria are these DNA got replicated, so they took the sample after 20 minutes. That is sample number 1, sample number 2. And then he took another sample in which he got the sample after 40 minutes. So that is the sample of the third sample. And this is actually the double of this 20 minutes. So it means the second sample is after the double time of first, and third is the double time of second. So after taking this one, just he dissolved DNA samples into cesium chloride. And cesium chloride, first time they used the chemical cesium chloride solution for a centrifugation. So that is called a density gradient centrifugation, in that DNA was settled down at their as their density. So when they spun at a very high speed in an ultra centrifuge for many hours, so the cesium and chloride ions tend to be pushed like this centrifugation machine. So solution centrifuged at this rate for 48 hours. So cesium and chloride ions were established in the tube at the bottom. So molecules of DNA were settled down and formed sediment to the level of their appropriate densities in the test tubes after just some time, when they just give the sample to some rest. So you can see here, and as well as we will see the next step. So this is called equilibrium density gradient centrifugation, where they use cesium chloride and for 50 to 60 hours at 100,000 times, more rounds, and results in the generation of the gradient of cesium chloride and banding of DNA. You can see the cesium chloride sample, and in that, with increasing of the density, they are settled down. Increasing the density means if they are having high density, so they will settle down, and then gradually they will move up. So in the sample, you can, as a result, that was proved that they are semiconductor model of DNA replication. How? So in the first sample, first sample is the control group, to just compare with that. So in N15 containing medium, all the DNA, because the replicated and the old one, both of the DNA were heavy. You can see here in the band. Continue growing first generation in N14 medium. So when they took at the 0 minute, so it is N15, all N15, because they didn't give any time to replicate the bacteria in N14. So they took at the 0 minutes. Then he took after replication cycle 2. So what happened? Heavy and light hybrid. Like after 20 minutes, he got a hybrid DNA over there, where the light and heavy both of the samples were there, because from N15 now they are replicating, but when they are replicating, they are taking N14 from the medium. So continuing growing, and replication 3, when he took another sample, so for more time, so he just got another he gave another replication, like where the DNA N14 replicated. So N14 replicated N14 again, not N15. Here, N15, it is replicated into N14 again, because that is continuously just present in N14. And then if more replication is done, so you'll see N14, N14, these three, this one, and this one more sample is just left, that is N15 and N14. So it means you can see the result here. Okay, so in a density, then when there is N15, so they are heavier. Heavier means they are 100% N15 DNA is present. It means when they replicated, all the DNA are N15. But gradually at 40, at 20 minutes, at 40 minutes, 60 minutes, and 80 minutes, you can see the results. So at 20 minutes, then they saw that like this is the hybrid line, and here N15 and N14 both are half and half, like 50% 100% replication is there. So N15 is replicated into N14, so that band represents in the center. At 40 minutes, when he saw that some of the hybrid, like where the N15 and 14 were present, and some are now getting N14. So like that, hybridization or hybrid DNA going to be less and less, and then there is N14, all N14 are representing. It means this DNA strand is having N14, and this DNA strand is having N15. So now it is proved after the experiment that all the DNA are replicating in our cells in semi-conservative manner. Like one is the old strand, and from there, that old strand that acts as template, and the new strands of DNA is going to be formed. So that proved a very, very important experiment regarding the DNA replication, that is semi-conservative replication. Dear student, that's it for today. Inshallah, we will talk about the mechanism of replication in detail tomorrow, inshallah. Allah Hafiz, wish you all the best.